All right, so let's say we have this structure right here, and then we were to have our our unsaturated fatty acid. So you see how there's just like less kind of overlap, like there's less potential intermolecular forces, and for that reason, they're easily able to be broken apart, which means that they need like lower temperatures in order for them to convert either from a solid to a liquid or liquid to a gas. And so your unsaturated are typically going to be found as a liquid, like your oils are going to be unsaturated. Your peanut butter, like very, very, what's it called? Kind of oily, not really that solid, like I was gonna say mantequilla, like butter. But yeah, so that's something to note. So they can be used for different things, such as storing energy, as we said before. So like, let's say you had a big meal and you had a big a fatty meal, like with a bunch of, let's say, grease or something, whatever it happens to be. Then rather than kind of like waste those lipids, your body is going to store them as triglycerides in adipose cells. So your fatty acid is kind of like your monomer that you're working with right here. However, if you want to store it, like you don't want to store kind of individuals, you want to store it in packing, kind of like in glucose. You don't store individual glucose molecules, you store them as glycogen, right? As kind of like things that are more organized in case you want to access them. And so for lipids, it's not going to be glycogen. It's actually going to be called triglycerides. And so what a triglyceride is as it sounds, it's a glycerol group. Sorry, it's a glycerol group. A glycerol group is this group right here, 3-carbon structure or 3-carbon carbohydrate, like this, except that the OHs are removed and they are exchanged with acyl groups. So this is an acyl group right here. Just whatever is mounted here, and then this is your acyl group. An acetyl group is this, like acetyl-CoA is just this. It's just a methyl right here. That's acetyl-CoA. This is an acyl group. So watch out for those two in case like it pops up in an exam. And then your triacyl glyceride, just like this. So these are, you're going to be your triacyl glycerides, your fats pretty much, and those are going to be stored in adipose tissue and adipose cells. Something to know about adipose cells, like I don't think you can get rid of adipose cells, they just shrink. So whenever you're dieting, it's like they're just shrinking, they're not decreasing in number. So it's kind of, that's why it's kind of easy to like go back and gain the weight than it is to lose the weight, because like it's easier to just fill in those cells and kind of just like take away the fat, something along those lines. I remember talking about biochemistry with Professor Dice, but besides the point. They can also serve as membrane lipids, as a phospholipid. So a phospholipid is a glycerol group with 2 fatty acids and a phosphate group on it. So this is your phospholipid, and your phospholipid is basically going to be within your cell membrane. So like, you know, these guys, like you have 2 fatty acid tails, right? And the reason why you have a polar head is because you got that phosphate group on there, and that phosphate group is going to have a charge. And the reason why I'm drawing it specifically like this is that like likes to interact with like in terms of hydrophobicity and hydrophilicity. And so these tails right here, these hydrogen carbon tails, they're non-polar. So they're all going to be sticking together. And then the polar head of your fats, like say like, you know, your carboxylic acid, if you were to deprotonate your carboxylic acid, what you will have is just a charge. So there will be like the polar heads. That's why your phospholipids are really useful for your membranes, because they kind of separate the cell from the outside environment. And additionally, something that you might be hearing is called a micelle. So micelle is just this kind of like form right here where you have like non-polar substances in a polar solution being kind of just like separated from the polar solution by using these fats. And if you're ever curious as to like why soap does what it do, is it just basically like missiles everything on your hand. Like, so whatever it is, like kind of like whatever kind of like lipidy fatty thing you have on your hand, you kind of like form those missiles, and then that's what takes it off. All right, then lastly, what lipids do is they make this structure called cholesterol. Cholesterol is like a kind of like a 4-ring structure, like 4 or 5-ring structure basically it just looks something like this. Like these are all like ring structures. And then from there, you want to be adding different kind of like methyl groups or functional groups. And that's what makes different kind of steroid hormones like testosterone or estrogen. So cholesterol is like one of the building blocks for those kinds of hormones. Let me just pull up what cholesterol looks like. Right here. Yep. So it's kind of like this 4-ring structure, and then they can add a bunch of different things to it to kind of just modify what it does, like testosterone, for example. Testosterone, holy shit, I suck at spelling. Yeah, so like the difference between these two is this one doesn't have this kind of like long non-polar kind of what's it called, branch on the side. It just looks like this, right? So lipids act as kind of the building blocks for these kinds of steroid hormones. So every steroid is just cholesterol, just modified into something else. All right, so lipids are net hydrophobic, aka lipophilic. So lipo means lipid, so it's lipophilic. So philic means to love, phobic means to be scared of. So you know, we do have this carboxylic acid end at the fatty acid, but overall it's going to be like, you know, that's just a carbon and 2 oxygens. Over here we have a bunch of carbons and a bunch of hydrogens. So overall it's going to be a hydrophobic substance, but it has the capacity, like in a polar solution, it's going to form this ring because it has the capacity, because it has those polar heads due to the carboxylic acids, or if you're talking about phospholipids, you've got that phosphate group there as well. So yeah, fatty acids are made of hydrophobic hydrocarbons and hydrophilic COH group, and they can be saturated or unsaturated. Okay, we talked about that. Any questions so far? We're about to get into the biochemical metabolic part. All right, so just to kind of like summarize what happens during lipid kind of digestion. So your triglycerides are going to be coming in from your small intestine, and they're going to be kind of emulsified / kind of be put into those missiles by the bile and by other things in your stomach or your small intestine, whatever it is, like wherever it is digested. Like, I'm sure Ashne talked a lot about that. But basically, in order to get into the cell, so like let's say this is like the lumen of where, like whatever it's resting on. I'm pretty sure it's like for lipids, it's like the small intestine. The small intestine typically absorbs most of the stuff. The stomach I think absorbs some proteins because it has like the enzyme pepsin in there. But for the most part, I think it's going to be in the small intestine, the jejunum, ileum, whichever one I forget. But so it's going to be, you know, say this is the lumen of the cell. So it's going to be, you know, found as the triacylglyceride. So in order for it to be introduced into the cell, it needs to be broken down by lipase. Lipase is just, you know, lipid ase is going to be the enzyme that kind of breaks down fatty acids. So lipase is going to break this down into its components, glycerol and the 3 fatty acids. And once it goes into there, it's going to be, after it enters there, it's going to be reconstructed into a triglyceride. And then they're going to enter the chylomicrons. Those chylomicrons are just bundles of fats and proteins, aka a lipoprotein, and they're going to be sent out into the lymphatic system. And so the lymphatic vessel, I believe that's next to the cells of the small intestine, are the lacteal lymphatic vessels. So once these triglycerides are broken down in the lumen and then enter into small components here in the enterocyte, once it enters the enterocyte, it's going to be reconstructed again as a triglyceride. Once it's reconstructed again as a triglyceride, it's going to enter the chylomicron and be kind of like a big ball of just fats and lipids. It's like fats, lipids, and proteins. And then after that, it's going to go into the lacteal lymphatic vessel and then go into the lymphatic system. From the lymphatic system, the chylomicron is going to be floating around doing its thing, and it's going to eventually find a capillary. What's it called? Capillary. And once it finds the capillary has like receptors on its like membrane that kind of recognizes the chylomicron. Either it's going to recognize the chylomicron or things like glucagon, things that kind of like break down storage molecules like glucagon or epinephrine. they're going to like tell the chylomicron to kind of just release their substances into the cells so that the cells can hold on to like those things. And the chylomicrons are just going to filter back and go back to where they were in the enterocyte of the stomach. All right, so triglycerides are cleaved back into these by chylomicron lipase, turned on by the chylomicron receptors or insulin, and sorry, I said glucagon. I meant insulin, like the storage of the storage of the triglycerides into the cells. Yeah, like the glucagon would be more of like breaking it down and sending it to the liver so it can be beta oxidized. Like beta oxidation and fatty acid synthesis all occur in the liver. So if you're just floating around in like chylomicron and just in a random capillary, and let's say insulin happened, like let's say you just, yeah, it all makes sense now. Like let's say you just had a big meal and like your insulin is like being pumped out. So basically your body's telling it like, okay, we have a lot of glucose in the blood, time to like, kind of like put that all the way. And so once your chylomicron is like floating around the capillary, the insulin is going to like basically tell the chylomicron to basically allow everything to enter the cell, and then it's going to be stored into the cell. All right, so so what's cool about the well, actually it's probably not that cool, but what's interesting about about fats is that they don't have this kind of solivation shell around them. So if you guys remember from chemistry, things like Na plus or potassium, or let's say like Cl minus, and once they enter the like let's say a polar kind of solution, they're going to be surrounded, let's say water, they're going to be surrounded by hydrogen, oxygen. They're going to be surrounded by these water molecules because they have, or actually for chlorine, let's just put NaCl, Na plus, because the oxygens are going to be attracted to the positive. So from here, they're going to create a solivation cell, basically just entrapping the solutes within the solution. And that happens if you're a polar molecule, right? You're just going to be attracted to polar things. However, because the fats are non-polar, the reason why like a pound, like the reason why like let's say a handful of fat and a handful of let's say proteins or glycogen, the reason why the fats are just like kind of less, like they carry less weight, is because of this just like water kind of sweater that they don't have. So like things like glycogen or protein, they do have this kind of solivation show like happening, so kind of like that water weight or whatever. And then the fats, they don't have that. So like over here, I have some statistics, like basically for a gram of glycogen, you can get 4 kilograms of energy. For proteins, like the same thing, but for fats, for a single gram, you could get 9 cal, because it's just like more invested within a gram. Also because of what we talked about before, which is a single fatty acid can produce many acetyl-CoA, and a single glucose, on the other hand, can only produce 2 acetyl-CoA. Okay. Any questions thus far? None. Let's keep going. Yeah, I'm pretty sure we're gonna finish maybe within the hour. Not really sure, but we'll see. All right, so now we're gonna go into fatty acid oxidation. Okay, so basically from the blood, the albumin is going to be transporting the fatty acids from the adipose tissue to where it's broken down into liver. So things like glucagon, they're going to want the breakage of the stored molecules such as glycogen or fats. And so you're going to have the kind of production of acetyl-CoA from your fats only occurring in the liver. So your liver is kind of like the kind of the blood glucose monster, like it's the one that regulates everything. And so the albumin is what's responsible for transporting that. So like let's say you have like just fat on your body, and so rather than the fat being like oxidized right there, it's going to be broken down. The albumin is going to be transporting whatever is broken down, whatever is necessary, to your liver, and then your liver is going to be the one that's going to be constructing the acetyl-CoA from the fatty acid. Okay, so in order for you to first start beta oxidation, you need to activate your fatty acid. So you start off with only just your carboxylic acid group and your acyl group and your carboxylic acid, and then your hydrocarbon at the end. This isn't going to be able to enter the mitochondria. And the mitochondrial matrix is where the mitochondrial matrix in the liver is where beta oxidation or fatty acid oxidation occurs. And so in order for this structure to be able to enter the mitochondrial matrix, you first need to activate it. And that's done by this enzyme right here, acyl-CoA synthetase. So whenever you see that at the ending, the synthetase, you can kind of assume that ATP is going to be involved. Like the tase at the end typically indicates that an ATP is going to be either produced or used in some capacity. And so in this case, the usage is we're going to be introducing a coenzyme. We're going to be using 2 ATP, producing ADP at the end, and then we're going to be basically attaching our coenzyme to our structure here. Okay, so this is the net result. We're going to have our fatty acid, then we're going to have the production of our our coenzyme, our acyl coenzyme. So again, as I mentioned before, the acyl group is this group right here. The acetyl group is this group right here. Just a methyl right there, like such. So we're going to have our acyl coenzyme A produced after so we're going to have the activation of our fatty acid into a acyl coenzyme. Once the acyl coenzyme is produced and it's become activated, it's able to enter the mitochondrial matrix and beta oxidation can occur. And so the outer membrane of the mitochondria has kind of like these pores that allow basically things to enter. So let's say we were to draw our cristae here. Cristae has a bunch of folding, and then you have our outer membrane. So basically the outer membrane has some holes where we can introduce some fatty acids. However, cristae is a bit more kind of like, I guess, strict in terms of transport. And so the reason why we need to activate it is not necessarily because of the transport from the cytoplasm to the intermembrane space. Like this would be this part right here would be the intermembrane space. So this, like the fatty acid going from here to here, that's not a problem. It's a fatty acid entering into the mitochondrial matrix where beta oxidation actually occurs. Because why do you guys think? So it's always good to like understand rather than to memorize. So why do you guys think that beta oxidation occurs in the mitochondrial matrix and not elsewhere? So you just have to think of like what is produced during beta oxidation, what is that location involved with, and kind of like the efficiency that cells have, like that we have as like animals. Like we want to be efficient, and so we kind of have like want things, if they're happening here, we're gonna also want things to happen there as well. So what happens in the mitochondrial matrix that makes sense why fatty acid oxidation would occur there.
Would that be the is it the Krebs cycle?
Yeah, the Krebs cycle or the tri. There's many names for the Krebs cycle, but basically, in the Krebs cycle, we have these acetyl-CoA. The acetyl-CoA is going to be introduced to oxaloacetate, forming citrate. Oh, it's called the citrate cycle. The citrate cycle. The tricarboxylic acid cycle. Because citrate is basically 3 carboxylic acids. But yeah, so citrate. Yeah, so basically the acetyl-CoA that is produced from fatty acid oxidation is going to be introduced in the acetyl-CoA from beta oxidation is going to be introduced to the Krebs cycle immediately in the mitochondrial matrix. So that makes things efficient, right? So it's perfect. If you're going to oxidize your fatty acid, might as well happen in the mitochondrial matrix. Okay, and if oxidation occurs in the mitochondrial matrix, the synthesis is going to be in the in the cytoplasm. And the reason being is it's just more efficient to just construct out here rather than inside here, because there's so many things going on within the mitochondrial matrix that constructing an entire fatty acid would just be better outside in the cytoplasm. All right, so basically, after our acetyl-CoA has been created, we're going to be using a translocase in order to introduce it to the mitochondrial matrix. So it's going to be called the acyl-carnitine translocase. The carnitine is going to make sense later on, because after you produce your acyl-CoA, you're going to be producing acyl-carnitine. Acyl-carnitine is going to have the shape needed for this enzyme, acyl-carnitine translocase, to like actually hold on to it and introduce it into mitochondrial matrix. So ACT is right here. This is ACT. So the acyl-CoA is going to come in and be introduced there. And then once it's inside of the mitochondrial matrix, the acyl-carnitine is going to lose its carnitine, and then the carnitine is going to go back up so it can keep being reused. Carnitine is basically just like a little bus fare. It's like, hey, like, or your bus ticket is like, hey, I have a trip to the mitochondrial matrix. So the coenzyme is like removed from the acyl. You introduce the carnitine, you introduce into the mitochondrial matrix, and the carnitine goes back up to the intermembrane space so that it can be used again for a different fatty acid. So that's your enzyme. And as I mentioned before, just like the ACT is going to be acting as a antiport. So I mentioned that the acyl carnitine is going to be going into the mitochondrial matrix, and what's going to be out of it. So this is what antiport is. Like one thing is going in one direction, the other thing is going the other direction. What's going to be going in the opposite direction is just carnitine, right? So the carnitine that's going to be reused in the intermitochondrial membrane space. Okay, so that's it for this slide. Any questions? Okay, none. So just overall, I'm going to have the products of fatty acid oxidation here at the bottom. So you're going to be producing one acetyl-CoA, one NADH, one FADH2 per beta oxidation turn. If you have a bunch of, if you have a huge hydrocarbon chain, you're going to be having multiple turns for beta oxidation, because a single turn is just going to get rid of 2 carbons from the fatty acid that you're working with. And so if you have a big one, you're going to have multiple turns. And so that's why we get so much energy from fatty acids, because per turn, we're producing a single acetyl-CoA, which is 10 ATP, and then one NADPH and one FADH2. So one NADH produces 2.5 ATP, one FADH2 produces 1.5 ATP. So that's already 4 ATP there. So from a single turn of beta oxidation, we're producing 14 ATP, which is kind of nuts, because there's still a bunch of other acetyl-CoA and NADHs and FADHs to be produced from our fatty acid. Okay, and lastly, the beta oxidation cannot help to contribute in gluconeogenesis, simply because, like, if you guys have ever heard this term, you can turn sugars into fats, but you can't turn fats into sugars. So what does that basically say? So sugars, like say glucose, can turn into acetyl-CoA, right? This is through the pyruvate dehydrogenase complex and also glycolysis. And then from acetyl-CoA, you can have fatty acid synthesis. And that's how you can make sugars into fats. However, you can't make fats into sugars. You can't have fatty acids being produce acetyl-CoAs. And you cannot have the production of glucose through gluconeogenesis, simply because this process of acetyl-CoA formation by pyruvate dehydrogenase complex. So pyruvate dehydrogenase complex takes pyruvate, the product of glycolysis, and produces acetyl-CoA. This pyruvate dehydrogenase complex is not a reversible step. So acetyl-CoA cannot be formed into directly into glucose through gluconeogenesis. Rather, it's going to have to be introduced as a different intermediate, let's say an intermediate from the Krebs cycle, like oxaloacetate. And once it becomes oxaloacetate, then oxaloacetate can go in and take part in gluconeogenesis. Okay. Any questions thus far? If not, we can keep going. Okay, here we have our first pathway. We have fatty acid oxidation. All right, so as I mentioned before, the fatty acid, let's draw our fatty acid right here. Our fatty acid has to get activated first. So can anyone tell me why the fatty acid has to get activated?
Is it so that it can pass into the mitochondrial matrix?
Mm-hmm. So first it's a fatty acid, then it becomes acyl-CoA. Once it becomes acyl-CoA, it has the capacity to become acyl-carnitine. Acyl-carnitine can then enter the mitochondrial matrix, right? So here, we showed it in the step before in the previous slide. up over here, we saw a fatty acid being synthesized into acyl-CoA by acyl-CoA synthetase, again, aptly named, thank the Lord, right? So here we have the production of acyl-CoA. So let me just try right there. CoA. And then from the acyl-CoA, we're going to use the enzyme carnitine acyl transferase 1, aka CAT1, to produce acyl-carnitine. So once acyl-carnitine is produced, it's just going to go straight into the mitochondria through this enzyme that we talked about before, the acyl-carnitine translocase. However, once the acyl-carnitine is produced, that's basically its fate, right? It's going to be introduced into the mitochondrial matrix, and it's also kind of just telling the cell that this fatty acid is going to be producing energy. And so you don't want to kind of waste fatty acids. Like you don't want to keep kind of beta oxidizing, because then you're just going to be wasting resources, as we mentioned before. So this is going to be the rate limiting step. It's going to be inhibited by a structure called malonyl CoA, which is important in fatty acid synthesis. So it's kind of like it makes sense, like for something that is important in fatty acid synthesis to inhibit fatty acid oxidation, because you don't want both of those things going at the same time, because it's just a waste of resources. Again, like fatty acid oxidation breaks down fatty acids, fatty acid synthesis produces fatty acids from acetyl-CoA, and so you don't want those two running at the same time. So one of the products of fatty acid synthesis is going to be inhibiting the rate limiting step of fatty acid oxidation, which is this CAT1, the carnitine acyl transferase 1. Okay, then you produce your acyl-carnitine, use acyl-carnitine translocase to kind of transport your acyl-carnitine into the mitochondrial matrix. Your acyl-carnitine is now introduced into the mitochondrial matrix. You're just going to remove that tag that says, hey, I belong here. So you remove the carnitine, you put it back up into the intermembrane space, so it can kind of like help facilitate the process of another fatty acid into the mitochondrial matrix. And you're going to do that, you're going to remove the carnitine by using CAT, but CAT version number 2. So and before we used CAT, we introduced the carnitine. That's something I forgot to draw. Carnitine goes in here, CoA leaves. And over here for CAT2, the CoA is going to be introduced and the carnitine is going to leave to produce acyl-CoA. So that's just to get into the mitochondrial matrix. Here we have an activated fatty acid. I don't know if I drew that just now, but all right, so now we have your acyl-CoA. After your acyl-CoA, you're just going to have 4 steps, and then you're going to be repeating these 4 steps every single time you need a turn for the fatty acid oxidation. So let's say, for example, you have this acyl-CoA over here. In this acyl-CoA, you're going to be first using FADH to oxidize this group right here. So if this thing is going to be oxidized, FADH2 is going to be reduced, and you're going to be producing FADH2 from FAD. And as I mentioned before, FAD is going to be involved with carbon-carbon double bonds. NADH is going to be responsible for carbon-oxygen double bonds, so or reactions. So here we're going to use, again, aptly named enzyme acyl-CoA dehydrogenase. Remember, dehydrogenase means that you're going to have some sort of redox reaction, and they're going to be producing enoyl-CoA, right? So enoyl-CoA, enoyl sounds familiar to that term, the enoyl, which was basically a kind of structure where we had the double bond and the what's it called? The oxygen close by. So just kind of like a reminder there, like we have the double bond close to this ketone group. Okay, that's how you can kind of like remember enoyl. So for enoyl-CoA, we're going to next use enoyl-CoA hydratase. So you're going to be introducing a water molecule. The water molecule is going to split off into an OH group and a hydrogen group. The OH group is going to go on the further carbon on the double bond. So right here, this carbon right there. And this hydrogen is going to go on this carbon over here to fulfill the octet that this carbon needs. So that's enoyl-CoA hydratase. After that, so you're using up water molecules in fatty acid oxidation. So next we have the L3 hydroxyacyl-CoA. So it's 1, 2, 3 carbons away. That's why it's called L3. L is just indicating the direction in which the OH group is pointing. So the L3 hydroxyacyl-CoA is then going to be oxidized by NAD+. If you're oxidizing L3 hydroxyacyl-CoA, you're going to be reducing NAD+, so you're producing NADH. So that's the 2 electron carriers produced in this pathway. Remember, you're going to be producing 1 NADH, 1 FADH2 per turn of the cycle. So once you oxidize this group with L3 hydroxyacyl-CoA dehydrogenase, again, super awesomely named, you're going to be producing your ketone, right? So you have a ketone from your OH group. Basically, this hydrogen was snatched off and the double bond occurred here. And that's what you have here. This resulting structure is just a beta ketone. Beta ketone just means that you have a carbonyl group, and then alpha and beta carbon, you have another ketone, so as a beta ketone. You're going to be using beta ketone thiolase to introduce a coenzyme A molecule. And after you introduce that coenzyme A molecule, it's just going to come in and bind over here. Once that comes in and binds over there, you're going to have the release of this structure, and that'll be your acetyl-CoA. So you would have something like so. At the end. So you have your acyl-CoA, it's just 2 carbons shorter, and then you have your acetyl-CoA, which is just your acetyl-CoA. And so like if you have let's say you have a 16 carbon fat T acid. You say you have this many carbons in your fatty acid. How many acetyl-CoAs potentially can you derive from this fatty acid? So you have 16 carbons. You can get 2 carbons per acetyl-CoA.
So 8 acetyl-CoA?
Yep, 8 acetyl-CoA. And how many turns would that take?
Is it 7?
Yes, 7 turns. Simply because at the end, the only thing that you're going to be left with is this structure right here. So once you have this structure right here, you don't need you don't need 2 turns to break this structure apart. Even though you have 2 carbons, even though you have 4 carbons, you would think, oh, 4 carbons, that's 2 acetyl-CoA, that takes 2 turns. It just takes 1 turn. You just have to split it off here. So that's why whenever you're trying to decipher how many turns you need, all you have to do is figure out how many acetyl-CoA you're going to be producing. After you know that much, all you have to do is subtract that number by 1. So in 16 carbon fatty acids, you're producing 8 acetyl-CoAs. So the number of turns that you need is just 8 minus 1, or 7. So now something interesting happens when you have kind of like an odd situation in terms of your fatty acid. So something that you guys saw in your full length was a monosaturated fatty acid. So in a monosaturated fatty acid, you have a group that you just basically have a double bond group, right? So if you have a double bond group already, the cell is efficient. It's not going to waste its resources. One of the steps in this process are going to be skipped over for that particular turn right here when we're tackling this double bond. Can anyone tell me which process is not going to happen if a double bond already exists? Which enzyme is not going to be used?
Is it the DH, the dehydrogenase?
Which one?
The first one.
Yes, the acyl-CoA dehydrogenase, which means if you have a double bond already present on your fatty acid, you're going to be producing less ATP per molecule because you're not going to be producing as many FADH as you would theoretically if you had a saturated fatty molecule. So that's one of the kind of random situations that you can have for your fatty acid. Another random situation that you can have is when you have an odd number of carbons on your fatty acid. So 16 is a nice and easy even number. However, let's say you have like 9, or let's say you have like 11 carbons in your fatty acid. And so what you're going to do is you're going to be tossing out 3 carbons at the end. So let's say you have 11 carbons. So the closest even number to that's makes sense is 10 carbons. You're not going to have 10. You're not going to have 12 carbons like from 11 carbons. So you're going to have 10 carbons. Those 10 carbons can be split off into 5 acetyl-CoA. And how many turns do you guys believe you're going to have if you're just going to be tossing out the, what's it called? The remainder carbons. Actually, give me a second. Actually, I have to double check with my notes real quick, but give me a second. Sorry about that, guys. I always get this one up confused. So like you have to take care of like the kind of miscellaneous ones where you have the what's it called? When you have a double bond already present on your structure, or when you have an odd number. So when you have an odd number, let's see. Word. Yeah. So rather than, so yeah, so you just have to remember for the odd number of, so I had it right the first time, I just kind of messed up over here. So if you have an odd number of fatty acids, so if you have an odd number of carbons in your fatty acid group, what you have to do is just subtract 3. So, okay, let's start over from here. So let's say you have 11 carbons. You have to subtract 3 carbons because those are going to be the 3 that you toss away. Like you're not going to be using those 3 at the end. And so you subtract 3, you're going to be using 8 carbons for the production of acetyl-CoA. So from 11 carbon fatty acid, what's going to happen is that you're going to be producing actually only 4 acetyl-CoA. And you're going to be tossing out the 3 at the end. And so you're only going to be producing 4 acetyl-CoA. How many turns do you believe that you're going to need?
Would you need 3 turns?
For this one, it's actually going to be the full 4 because you're not going to be crossing off at the end. You're not going to be kind of before yeah. So if like before you can just, if you had 4 remaining, you could just split it in half, and that's, it only takes 1 turn for 4 carbons. So that's why you had to subtract at the end. But if you're not going to use every single carbon, if you're going to throw out 3 carbons at the end, then all you have to do is the 4 turns for those 4 acetyl-CoA. And so at the end, you're just going to be left with this structure right here. And this structure right here is 1, 2, 3. Oh, shit to me. Say like this structure right here, like this is not enough to produce another acetyl-CoA. So this thing is just tossed out, and you're left with your 4 acetyl-CoA and 4 turns. Okay. Any questions about the mechanism? So like the biggest thing they're going to ask you for a problem like this is just like, all right, so we'll do a practice problem actually. So let's say they ask you, you have 12 carbons, you have a 12 carbon fatty acid, and it's not activated. Okay, they want to see how many ATP you're going to be producing from the oxidation of this fatty acid. They want to know how many acetyl-CoA you're going to be producing, how many turns, how many NADH, and how many, sorry. Yeah, my laptop is acting kind of wonky today. I think it's just been on for too long, so it's like overheating a bit. Yeah, actually a lot. And if I ever freeze, just kind of like text me on GroupMe because I'm not gonna know. And then FADH2. Okay, so I'll give you guys like a minute or two, and then we'll go over it. So you have 12 carbons, it's not activated. How many ATP you can produce, how many acetyl-CoA, how many turns in total, how many NADHs, and how many FADHs. And just in case you wanted to practice the odd number or like this exact example, I have it in my notes, but for now, just like practice it live so we can go over it. So whenever you finish, anyone can just volunteer and like and post up the numbers that they got. Sorry that I'd be forgetting some things sometimes. I'm kind of scatterbrained today. Oh, and I'm pretty sure we're going to finish before 4 o'clock. So if you have any questions, let's say from like past full lengths that you wanted some like clarity on, we can definitely do that as well. If not, you guys can join a different session, or we could just like talk about MCAT in general. Any questions, kind of like have a free open office hours that we finish early today. Kind of thought it'd take longer, but not too bad. I should just remember it's 12 carbons. It's not activated. Why is this important? And also tell me where you're making most of your ATP from. Like what cellular respiration pathway are you getting most of your ATP from? Right. Phos. Yes, but yes, no, you're correct. I kind of just said it wrong. Saying more like where do you get most of your NADH, your electron carriers? Where do you mostly get them from? Yep. The citric acid cycle. That's what I was trying to say before. I was blanking on it. It's like, oh, is the tricarboxylic acid cycle? That's citrate, and then just another citric acid cycle, and then the Krebs cycle, because Krebs was the homie who found it. And they're just like, well, we can't give him too much credit, so they're trying to say that the citric acid cycle, and then someone thought it was kind of funny that citric acid has like 3 carboxylic acid groups, so it's like the tricarboxylic acid as well, TCA. Okay, anyone figured out how many ATP, acetyl-CoA turns, total number of NADH, total number of FADH produced?
Can I attempt to complete it?
Heck yeah.
All right, I'll go from the bottom up. FADH2s, in total, I got 11. NADHs, I got 23.
Okay.
Rounds, I got 5.
Yes, 5 turns. So we'll do the individual maths, like where we get the stuff from. So right now I'm just saying, but we'll do it individually, like later on.
Word. Acetyl-CoAs, I got 6.
Yes.
And then ATPs, I was a little iffy. Because I know at the end you like normally do, I think, like -2 of your full total, but my like total was 80 ATP. And yeah.
So why do you? So Emmanuel, were you here from the beginning?
No, I wasn't.
Okay, so we went over a spot where we needed an enzyme, I believe it was acyl-CoA synthetase, that required ATP to activate our fatty acid, so the fatty acid can be turned into coenzyme. So acyl coenzyme, and then the acyl coenzyme turned into creatine, or to acyl creatine, or not creatine. What am I saying? Acyl carnitine, and then it enters the mitochondrial matrix. It takes 2 ATP for those fatty acids to be activated. And so if it's not activated, like if they just give you a fatty acid, at the end you subtract 2 because it takes 2 ATP to actually activate it. If I were to be like, oh, like here's a 18 carbon acyl CoA group, then you don't have to subtract 2 at the end, because like the what's it called? They're asking you about the what's it called? The acyl group. So the acyl CoA group, which means that they kind of skipped over the activation part.
Word. So like when it's like not like activated, you would like subtract 2 when it's not activated. Okay. Thank you.
Yes. Okay, so we have 5 terms, which means we're going to be producing 5 NADHs from those turns, 5 FADHs from those turns, H2s. We're going to have 6 acetyl-CoA. And then from the acetyl-CoA, we're going to be producing 6 ATP or GTP. We're going to be producing 3 NADH per acetyl-CoA, so 3 times 6, 18 NADH. And then for FADH, we produce 1 per turn, so it's going to be 6 FADH2. BH2. So 11, yep. 5 plus 18 is 23. And so, yeah, these are the numbers. And then you guys answered my questions. Where is NADH mostly going to be formed? It's going to mostly be formed in the Krebs cycle. And your FADH is mostly in your Krebs cycle as well. And yeah, that's basically all I really need to know for like fatty acid oxidation. Sometimes they ask you about, what's it called? Like what resources you need for fatty acid synthesis, but this one's going to be most likely the form that they ask you for like fatty acid metabolism in general. Is there any questions for this problem? Like this will happen, like you will see this problem like maybe like once every other exam, I would say.
Yeah, I have a question. So how many ATPs do you get per NADH and per FADH2?
You get 2.5 ATP per NADH. Do you know the theory why there's decimals for these ATP, for these numbers?
No, I don't really remember.
So since we have extra time, like we're going to finish early, might as well explain that. Because if you have a question about it, I'm sure other people have questions about it. That tends to be the case for what's it called, for MCAT in general. So we have these structures here. This would be our electron transport chain. This is our complexes. This is our ATP synthesizing monster. And so what happens with NADH is that, it's an electron carrier, is going to be donating its electrons. So NADH donating its electron to form NAD plus. Here, these electrons, as the name states, is an electron carrier, the electron transport chain. So electrons are transported here, 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 here eventually, and then it reaches oxygen. If you have a bad number of electrons to your oxygen, you're going to create what? What was the thing that happens if you kind of give oxygen a bad number of electrons? You guys remember what that was? We talked about it earlier today.
Creates a radical, did you say?
Yes, a radical. And then what's, glutathione and then NADPH. Okay. So these electrons are negatively charged. They're going to be attracting protons from your mitochondrial matrix. Remember, this is the mitochondrial matrix side. This is the intermembrane space. And so those protons are going to be like, wow, those electrons moving down that line, awesome. And so they're going to be pumped up here, creating a proton gradient. And so the reason why we have these kind of decimals here is because this complex pumps out 4 protons. So 4. This one pumps out 4 protons. This complex doesn't pump out anything. This complex doesn't pump out anything. This one pumps out 2 protons, right? Because the electrons are starting to lose their energy as they go down. And so at the end, when you're donating an electron in this carrier, you're going to be producing 10 protons, right? 10 protons. They found out when protons enter our ATP synthesizing enzyme is that it's kind of like a water mill where like the water rushes down and it cranks. So what the ATP enzyme does is that it takes a phosphate group, an inorganic phosphate group, and it takes an ATP. And with that cranking, it kind of just squeezes the two together like physically, and you produce your ATP. And it takes 4 protons in order for that thing to twist completely. And so if you're producing 10 protons from a single NADH, you divide that by 4 protons because you need 4 protons per ATP, you get 2.5. So that's why you get 2.5 ATP from an NADH. Can anyone tell me why FADH produces less?
It doesn't pass through the first electron receiver.
It donates its protons, sorry, not its protons, its electrons over here. So it completely misses its first complex. The electrons are introduced here, blah, 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 they go down. So they only shuttle out 6 protons. You divide 6 by 4, you get 1.5. And just in case you guys might have missed it from whatever lesson I taught it in, cytoplasmic NADH is also going to be sending its its electrons to this second complex rather than the first, simply because they have to go from the, glycolysis happens in the cytoplasm. When the NADH is coming in from the cytoplasm to the mitochondrial matrix for the ETC, it loses some of the electrons, basically lose some of this potency. It's not that it loses this potency. I'm not exactly sure why, but the NADH is going to be introducing its electrons to the second complex rather than the first. And so for that reason, funnily enough, prokaryotes actually produce more ATP than us when we're talking about ATP production from cellular respiration, because our NADH produced in the cytoplasm from glycolysis kind of is introduced as an FADH almost. And for the bacteria or whatever prokaryote, their electron transport chain are on their plasma membrane. So they, everything is kind of in the same spot. So they don't have that kind of weird situation happening. Okay, and I think I was going to explain something else. Is that it? Does anyone have any questions? Oh, and then FADH just goes here, divide that. Any questions? If not, we can continue with our lesson. All right, so here we got fatty acid synthesis. It's going to happen in the liver, except it's going to happen in the cytoplasm because we're taking acetyl-CoAs and we're going to be constructing a decently long molecule. And rather than do that in the really busy mitochondrial matrix, it's going to happen in the cytoplasm. And then once it's in the cytoplasm, it could be shuttled out and stored somewhere else in like an adipose tissue or whatever it is. But okay, so the acetyl-CoA cannot cross out of the membrane, but citrate can. And so you're going to infuse acetyl-CoA with a molecule to create citrate. And then once citrate is outside of the mitochondria, it releases the acetyl-CoA and the oxaloacetate that was embedded with the acetyl-CoA in the first place. So you take acetyl-CoA, you add oxaloacetate. Oxaloacetate is a 4-carbon structure, and you produce citrate. Once citrate leaves the mitochondria and it goes into the cytoplasm so that the acetyl-CoA can be released, you have that release. So let's see, and right here the split mitochondrial matrix, cytoplasm, acetyl-CoA plus oxaloacetate. Oxaloacetate does its thing, and actually during this process, this is called the citrate lyase cycle. It might occur in a, you might hear this pathway in a full length, you might not. What's kind of crazy for me for this pathway is that you actually produce a little bit of NADPH. So typically, if they ever ask you like, hey, is the pentose phosphate pathway the only place where you can produce NADPH? The answer is no. In the citrate lyase cycle, you can also produce NADPH because something got oxidized. All right, so yep, NADPH reduces a carbonyl group, make 2 carbons at a time. Thus, okay, yeah. So typically for fatty acid synthesis, you're going to be, acetyl-CoA is 2 carbons. You're going to be producing you can be producing your fatty acids 2 carbons at a time. And then, yeah, the citrate lyase cycle here at the bottom, I have my notes at the bottom. So like if you ever miss something that I say, it's all in my PowerPoints, and that's why I kind of like make my PowerPoints these kind of like very just dense. So because if like you miss out on something, I don't want you to like miss out on that information forever. It's going to be somewhere within the PowerPoint. Okay, so now talking about acetyl-CoA once it's in the what's it called? Once it's in the cytoplasm. So once you have your acetyl-CoA in the cytoplasm, what's going to happen is that you're going to be using the structure called an ACP. The ACP has these thiol groups. Basically, your acetyl-CoA and your mannonyl-CoA, something we talked about, the mannonyl-CoA, is going to be attaching to these groups in the ACP. So the coenzymes are going to be removed, and they're just going to be attaching to your groups. And what happens is that your bottom group right here is going to decarboxylate itself. Once it decarboxylates itself, it produces a decently good nucleophile, and then that is what's going to kind of drag this this top ketone group right here, your acetyl group, to the bottom, and then that's how your fatty acid extends pretty much. So you load on the acetyl-CoA, you load on mannonyl-CoA, and you produce these two structures. After that, you're going to get some decarboxylation, producing a decently good nucleophile. From that nucleophile, it's going to attack over here. Once it attacks over here, you're going to be extending this chain until you keep extending it, keep reducing this carbonyl group until kind of like it's flat. So you just have that hydrocarbon chain, and that's how you produce your fatty acids from acetyl-CoA. So let's go through the mechanism. So the first things first is to load the ACP. So you take acetyl-CoA and you use acetyl-CoA carboxylase. And the carboxylase is just going to be adding a carboxyl group to your acetyl-CoA. So that's something interesting, right? We've always been kind of just seeing the carboxyl group kind of come off of the molecule, but in this case, we're actually introducing the the carbon dioxide group. And because of that, it's not really spontaneous. You have to use an ATP. So using ATP here, you produce ADP, and you have this structure here, a 3-carbon structure, 1, 2, 3. You have malonyl, I can never say the name, malonyl. You have malonyl-CoA. This is the molecule that I talked to you guys about before. This is the molecule that's going to be inhibiting fatty acid oxidation. So just that, this is just to make sure that the two pathways don't kind of happen at the same time, right? Like it would suck if you're just producing fats and then in the same second, a different pathway is just using it all up to produce, kind of like what you just started with. So this malonyl-CoA from your fatty acid synthesis is going to be inhibiting that beta oxidation. Okay, so you take malonyl-CoA, and then you use this aptly named enzyme, malonyl transacylase. So trans means to just to change it, and then the acylase is to change the acyl group location. And so you're going to be taking off the CoA, and you're going to be introducing it to the ACP, producing this mannonyl ACP, right? So the mannonyl ACP mannonyl is now loaded onto your ACP. Next, you just have to load on your acetyl group. And so you're going to take your acetyl group. You're going to be using acetyl-CoA transacylase. Again, you're just going to be moving the acetyl-CoA group to your ACP. You're going to be, I should have drawn that. You're going to be removing your CoA. No, did I draw that wrong? I believe I did. So it's just like this. This is not here, like that. So basically the thiol group that was once a part of the CoA is now attached to the acetyl group. So the CoA leaves, it bounces, and then you have your acetyl ACP. All right, so again, I have to erase that part over here. This is kind of redundant, having a sulfur bind to a sulfur. All right, so I'm gonna update these slides, and that part's fine. Okay. So yeah, just note that if I had the if you see in your slides the kind of CoA attached at the end, just kind of modify, erase it, or if you can, just like erase it off. If you can't erase it off, there's an option to kind of like draw on it, so you could just like kind of whiten it out with some white color. Okay, so now you have the acetyl-CoA and you have the malonyl both attached to your ACP, and now it's loaded. So the first thing, as we mentioned before, is just going to decarboxylate it with the specific enzyme. To typically the enzyme is going to be beta keto acyl synthase. And what's going to happen is the carboxylic acid group is going to leave. So that carboxylic acid group that we initially had for malonyl to produce malonyl is going to bounce. And then you're going to be introducing our acetyl-CoA, because remember, as we said before, for fatty acid synthesis, you're mainly going to be producing even number of fatty acids, like even kind of number of carbons of fatty acids. So then malonyl was 3 carbons because we had acetyl-CoA, which is 2 carbons, in addition to the carbon dioxide as 3. And so just recall that we're going to be producing even numbers. So the carbon dioxide just bounces off so we can have our even numbers. Okay. Any questions so far about the loading of the ACP or anything like that? All right, if not, we can keep going. So really quickly, as we mentioned before, there is a rate limiting step for beta oxidation. There's also going to be a rate limiting step for fatty acid synthesis. And of course, it's going to be at this kind of nuts looking enzyme over here at the acetyl-CoA carboxylase, that the thing that's introducing this carboxin, this carbon dioxide group. So in this enzyme, we have 2 things that activate it and 2 things that inhibit it. The 2 things that are going to be activating it is citrate and insulin. Can anyone tell me why citrate or insulin would be activating fatty acid synthesis? So think about it before. How did we get acetyl-CoA to be in the cytoplasm in the first place? And what does insulin do for storage molecules? Right. And if someone wants to go, they can go. If not, I'm just gonna have to snipe someone real quick. All right. Oh, see some in the chat. Insulin is used to store glucose. That is facts. Insulin is also used to store things in general. So if you have, like, let's say you had a big meal, you're gonna have a lot of insulin going through your bloodstream. It's going to be telling the glucose in the bloodstream to enter the cell and be stored as glycogen if you're in the muscle, if you're in the liver. And then the same thing is going to be true for your fatty acids, right? If you have enough kind of energy because you just had a big meal, then the insulin is going to be like, okay, we really don't need all these fatty acids to be broken down or whatever. Let's kind of let's take these acetyl-CoAs, for example. Also, like, let's see if you have a bunch of acetyl-CoAs just floating around because you had a bunch of glycolysis, Krebs cycle, and all that, or you had the glycolysis of pyruvate dehydrogenase. If you had all that done, then you're gonna have a bunch of acetyl-CoA just floating around. And so rather than just have that, you're going to be producing fatty acids to be stored as triglycerides. So citrate, so that explains insulin. Insulin kind of like stored, like it increases the size of storage molecules such as triglycerides or glycogen. And then for your citrate, well, citrate is the thing that kind of transported the acetyl-CoA out from the mitochondrial matrix. So if you have a bunch of citrate, it's like, dang, we have a lot of kind of, I guess, like we have a bunch of customers. We have a bunch of citrate that has to donate their acetyl-CoA for the production of what's it called? For the production of fatty acids. All right, so next we have the inhibitors, which is long chain fatty acids and glucagon. Glucagon and insulin are antagonists. So if insulin is like, okay, storage molecules increase in your size, because we have too much, then okay, cool, that's what insulin does. Glucagon would be the opposite. They would tell glycogen and fatty acids to be like, hey, we've got a deficiency of glucose in the blood here, so let's kind of break stuff down. And so glucagon would inhibit fatty acid synthesis because we need the fatty acids to actually not be synthesized, rather to be oxidized, right? And then long chain fatty acids, excuse me. So for long chain fatty acids, these long chain fatty acids, they're going to be, if they're present, that means that there's, an abundance of them. If there's an abundance of them, you don't need to make more. So that's when you're probably got to tell your cell to transport the fatty acids to adipose tissue and via albumin, or just kind of, stop the process of fatty acid production, because, those are long molecules, not necessary to like make more of them if they're already present. All right. So after you've loaded your ACP, the next step would be to, The next step would be to remove the ketone from your ACP. All right, so let's say, for example, we have your ACP here, and it's loaded like such. And you have your two ketones, right? And so what you're going to have to do here, okay, so I just like drew it over here like the ACP. You can imagine like the bead of it, like right here. So what you're gonna have to do is just basically reduce the heck out of your ketone, so it could just be flat, like it could be just like a flat carbon group rather than a carbonyl group. So you're gonna be using NADPH. Again, just we're using NADPH for reductive power. So NADPH is not going to be used for the electron carrier, the electron transport chain. So the NADH is going to reduce the this here ketone to an alcohol group with the enzyme beta keto acyl reductase, producing your alcohol group here. After that, you're going to be using the enzyme 3-hydroxyacyl dehydrogenase to take your D3 hydroxybutyrol butyrol ACP and removing a water molecule. So it's kind of, if you find things similar, it's because things are similar. This is kind of reminding you of beta oxidation when we were kind of first introducing your double bond. After you introduce your double bond, you put a water molecule in it to make an alcohol group, and then you oxidize your alcohol group to make a ketone. It's basically just the opposite steps for fatty acid oxidation, right? Just taking and rather than synthesizing a ketone, you're just removing the ketone to form your straight carbon, your hydrocarbon, right? So after you make your alcohol group, you're going to form your double bond here by removing a H2O. So you're going to be moving this OH from this carbon. You're going to be moving a hydrogen from this carbon, producing this double bond over here. This is called a cratonyl ACP. I just remember that the ACP is just this structure right here. I'm just abbreviating it here and for the sake of space. And then you're going to be using your enoyl reductase to reduce this double bond with your NADPH, and you're going to be producing NADP plus at the end. So recall I mentioned that FADH is typically used for the reduction or oxidation of carbon bonds and rather than carbon carbon oxygen bonds. But just recall that NADPH is the powerhouse of reduction, and that they can also take care of the carbon-carbon double bond as well. So whenever you're thinking of the reduction of ketones during fatty acid synthesis, just make sure that you're using NADPHs rather than FADHs. Okay, any questions thus far? Okay, so if not, let's go through a quick exercise. So let's say we had, we're trying to produce a 16 carbon structure, trying to produce 16 carbon fatty acid. So just kind of tell me how many acetyl-CoA you're going to need. And don't cheat, like I know you guys can see the bottom, but like don't look at it if you want to practice. So how many acetyl-CoA you're going to be using? How many ATP you're going to need? How many NADPHs? And how many, what's it called? Yeah. Oh, and then so this is for your reactants or reactants. So how many you're going to need for these? And if you already saw the numbers down here, then just kind of explain to yourself why those numbers make sense, right? And then products, we're using our how many CoAs, how many ADPs, how many NADPHs, or sorry, how many NADP pluses, how many CO2 groups. And well, I mean, the CO2, not necessarily, because like you use CO2 in the beginning, you toss it out at the end. So it's like kind of net, doesn't really matter. And then how many water molecules you're going to be producing. Okay, we'll give this one like 5 minutes and then we'll discuss it and then we'll wrap it up with our, with our last slide. So we'll wait until 3:25 and then we'll discuss it. And then just lastly, how many turns? All right, about 2 more minutes left. Does anyone want to like start filling these in? I know you guys can annotate, so if you want, like you could just annotate the numbers in. I'll confirm them, and we'll discuss like why they might be wrong, or a little bit off. Why they might be wrong, why they might be right, or a little bit off. Jeez. There you go. Just going to close this. So that's gonna cheat. Ah, Jesus Christ. Yes. Okay, so it's time. It's 3:25. All right, so Ashley was correct. You're going to be needing 8 acetyl-CoAs for a 16 carbon fatty acid, simply because a single acetyl-CoA is 2 carbon groups. If you need 16 carbons, you're going to need 8 2 carbon groups to do that. 8 times 2, 16. All right, so we talked about how it's going to be, we talked about how we need to activate our, we need to add our we need to add our malonyl CoA to the ACP. And the way that we do that is by taking an acetyl CoA, adding a carboxylic acid group to it. That doesn't happen spontaneously, so it's going to take what's it called, an ATP to do that. And since we're only going to be using, it's like the same kind of thing with beta oxidation, where the number of acetyl CoAs, the number of turns that you need is just acetyl CoAs minus 1. So it's going to take 7 turns. In terms of ATP, if you're doing it 7 times, if you're doing malonyl, attachment 7 times, it's going to take 7 ATP. For NADPH, since you're reducing twice in this process, you're reducing when you're using beta keto acyl reductase and also ennoyl reductase, you're going to need double the amount of turns. So you're going to have 14 NADPHs. For CoAs, it's the same number that you have for acetyl CoA, so it'd be 8. For ADP, if you're using 7 ATP, you're going to be producing 7 ADP. Same deal with NADP plus, so 14. And then lastly, for every turn you have, you're going to have the extraction of a water molecule to produce their double bond. So you're going to have 7 H2O at the end. And then you have 7 turns. Okay, so any questions for fatty acid synthesis? If not, Ashley, do you mind erasing the 8 and the 7? Oh, thank you so much. Okay, so last 2 slides, and then we can call it a day. And we just have the the last session from 4 to 5, which whoever you can, whoever you want to join for that group. So the last thing we're going to talk about is ketogenesis. So whenever we're starved, let's say like we have low blood glucose and our glycogen stores are pretty much depleted. So how are we going to be using, how can we do cellular respiration if we don't have glucose or carbohydrates to kind of oxidize during cellular respiration? And so what you can do is you can take the fatty acids, you can break them down using fatty acid oxidation to produce acetyl-CoAs, and those acetyl-CoAs can go into the Krebs cycle and produce just a crap ton of ATP, right? However, where does the, what's it called, fatty acid oxidation occur? It occurs in the liver, and the liver isn't the only organ that needs these acetyl-CoAs. So how are you going to be transporting, let's say, these acetyl-CoAs to the brain or the central nervous system when the brain has something like the blood-brain barrier in which fatty acids can't even cross into, or acetyl-CoAs, right? So what you're going to need is a kind of vehicle to transport these acetyl-CoAs so that it can enter different places, and that's going to be in the form of ketone bodies. So the ketone bodies are going to be kind of different structures that have the capacity that once they arrive to their destination, have the capacity to convert back into acetyl-CoA so that it can enter the Krebs cycle and do its thing. So in the liver, where we have most of the blood glucose kind of magic happen, we're going to also have ketogenesis, which is the genesis or the creation of ketone bodies. We have 3 different ketone bodies. We have acetone, which is just a ketone with a methyl group, 2 methyl groups. We have acetoacetate, this structure right here, and then we have beta hydroxybutyrate, which is basically this alcohol which is oxidized into a ketone. And just recognize the language used, the beta hydroxybutyrate, the beta, it's just alpha beta, right? Just 2 carbons away, make something a beta. And so what's kind of, that, I mean, that's great and all. It's just like, oh wow, we can have like these why do we need carbohydrates if we can just use fats all the time? If a single fat molecule can get us way more ATP than a glucose molecule or just a random carbohydrate molecule, why aren't we using fats for all of our kind of needs? And so the reason is that ketone bodies are actually acidic when you introduce them, except for acetone. Acetone's okay, but even acetone, like, it's got the alpha hydrogens that can be potentially acidic. But as you can see here, the acetoacetate, and you have the beta hydroxybutyrate, if they're just free floating, because these ketone bodies, they're just going to be rolling out from the liver, if they're free floating, they have the capacity to become acidic. And so what happens if you do too much ketogenesis is that you have this thing called ketosis or ketoacidosis, which is basically your blood pH. If you're having an acid kind of circle, like same thing with lactic acid, right? If you have ketone bodies that are acidic, kind of just circulating around in your blood, then what's going to happen is you're going to have a decrease in your pH, which is in acidosis, it's a bad condition. This typically happens for people with diabetes, simply because they can't get the glucose that they eat from their meals into their cells to do cellular respiration, and so they typically use their fat stores in order to produce like enough energy for their system. And what happens is that they just have so much ketone bodies lying around that you just have an overall a decrease in pH. And so just some symptoms of ketoacidosis or ketosis: you have fatigue, you have confusion, and you have breath that smells like acetone, because acetone is one of the ketone bodies, and it's crazy. Like, some people got arrested before because when they blow into the breathalyzer, the acetone registers, or like they just like the breath registers as if they've been drinking. It's like not really it. They just had ketoacidosis. Ketogenesis is typically the last resort. You have no carbs left, or you do have carbs, but the thing is your carbs won't enter into your won't enter into your cells. Unfortunate event. You're gonna have hyperglycemia. If you have too much blood and if you have too much glucose in the blood. And what happens is that at the end, if you have an excess acetyl-CoA, then you're going to have basically just a negative feedback of the Krebs cycle, because the acetyl-CoA is produced in the fatty acid during fatty acid oxidation. Many of the enzymes of the Krebs cycle are inhibited by acetyl-CoA. So basically, if you have an excess of acetyl-CoA due to ketogenesis, your Krebs cycle is not going to be working properly because you have inhibition of it, while at the same time you have an excess of acetyl-CoA. Okay, any questions thus far? If not, we can just go through the mechanism and then we'll be done. We'll finish early, or if you guys have questions, feel free to just stay behind, or you can just join a different session, whatever it is. All right, so for the ketogenesis mechanism, what you have is you have your what's it called? You have your acetyl-CoA. You have two of them. You're going to be using a thiolase. Remember, in fatty acid oxidation, we're using a beta ketothiolase. Sorry, a beta thiolase. Was it beta ketothiolase or beta thiolase? Let me see. I think it was a beta ketothiolase. So in this case, we're just using a regular thiolase to combine, rather than to cut, to combine these two acetyl-CoA groups to create acetyl-acetyl-CoA. After that, we're going to be introducing another acetyl coenzyme to form HMG-CoA using the enzyme HMG-CoA synthase. So this CoA is just going to be plopped out, and then you're going to be producing this 6-carbon structure right here. After that, you're going to cut it up. You're going to be removing an acetyl-CoA. So it kind of sounds redundant, but at the end of the day, you're going to be producing this different structure that you had before. Right, the structure over here you had, and here you don't have any oxygens, so you don't have no kind of like a carboxylic acid. Here we do have a carboxylic acid. That's why HMG, like this whole process occurs. So from HMG-CoA, you use HMG lyase. Lyase is used to cut. The acetyl-CoA is going to be bouncing, and then you're going to be getting acetoacetate as one of your ketone bodies. So acetoacetate is used to form every other ketone body. So you're going to be using acetoacetate. If you randomly decarboxylate this acetoacetate, so like if you take this carbon and these two oxygens producing a carbon dioxide, what you would be left with, and if you oxidize this, what's it called? This OH group, what you'll be left with is an acetone. That's one of your ketone bodies. And then lastly, if you were to use beta hydroxybutyrate dehydrogenase, butyrate, remember butyrate means 4-carbon sugar, 4-carbon structure. So just recognize the name hydroxybutyrate. This is a hydroxybutyrate. It's beta 1, 2, 3. Alpha beta. This is a beta hydroxy. Hydroxy means alcohol. Hydroxybutyrate dehydrogenase. Using an NADH to reduce this structure. You should be using it to oxidize. So let me just draw the form in the proper way. I had it drawn oppositely actually. So if you're moving it from there to there, NAD plus to NADH, you're taking away this hydrogen and you're oxidizing this alcohol group to a ketone. And so if you're going to do the opposite direction, like you're taking, if you're going to take your created beta hydroxybutyrate and trying to form acetoacetate, you just have to do the opposite, right? You just have to go from this direction to this direction. So reducing your ketone rather than oxidizing it. And then lastly, when you get to your destination, wherever it is that you want to go, you just have to regenerate your acetyl-CoA. How do you do that? So for example, for acetoacetate, you can form acetoacetate from your other structures, from your acetone and from your, well, less likely from your acetone, but mainly from your beta hydroxybutyrate. So for the regeneration step, you're taking your acetoacetate and you're going to be using a CoA transferase. Succinyl-CoA is going to be introduced just to get the CoA, the coenzyme from the succinyl. The succinyl is just taken out. You've effectively added your coenzyme A to your acetoacetate, replacing the oxygen group here. And then once you have this structure over here, you have your acetoacetate CoA, which is basically the structure that we had initially. And after that, you're going to be using the same enzyme, thiolase or ketothiolase, in this case, to cut the, so nice. So in beta oxidation, we use ketothiolase to cut. We're using ketothiolase again to cut, and then I'm guessing to construct, we're using just thiolase, ketothiolase used to cut. Okay, so here, acetoacetate, using ketothiolase, you're introducing a coenzyme A, so you can get 2 acetyl-CoA, and that's how you reproduce your acetyl-CoAs from your from your ketone bodies whenever they arrive to their destination. But that's the end of fatty acid synthesis and ketogenesis, just 11 slides. Again, all of my slides are decently comprehensive. And so if you miss anything that I said, you can also always refer back to it. But for now, we have the session lasting until 4 o'clock. So we got like 20 minutes left. If anyone has any questions, feel free to ask. If not, you can have a mini study hall until a new session pops up, or you can join someone else's session. Like I say, that you're discussing some questions that you might want to look over. Definitely feel free to. All right, guys. See you later.
Thanks, bro.
You got it, man. All right, so switch some things around. Oh, I forgot this thing is still recording.